Well, I suppose we'll get started. We've got a bunch of students missing up the spring carousel today. So we're recording this class for them. So we'll see how it all comes together. So do our 5-minute break. Number 9 is due this Thursday. Other assignments, I think everybody's turned in their classroom pulse. The YouTube lecture 16 online had a question at the beginning. I needed you to answer me. I've only got about a third of the class. So get that answer to me at some point by email. And then we've got your question 3 was due last Thursday. So make sure that you get those in. Since I wasn't here, we'll extend those out. Exam 2 is a week from Thursday, covering lectures 10, 11, 12, 14, 15, 16, 17, and 18. So covering will break off somewhere in this Thursday that seems logical. We're coming up probably in 2 weeks to start talking about metabolism. When we're talking about metabolism, I'm going to use 4 different colors. So they match my pen here. We got black for, we're going to do parts and pathways, red for hormones, green for nutrients, and then blue ink for processes. So if you want to match the colors I've got, those are the colors I'll be using. You can use whatever colors you want, but those are the colors I'll be using. So we'll need those in about 2 weeks to get that done. So any questions or concerns? Everybody able to get through the YouTube for lecture 16? Works up on Brightspace? Any other questions or concerns? So letters in a sequence of heart events, given the set we have there, A through D, which event comes first? Signal from the AV node, signal from the SA node, the closing of the AV valves, or the opening of the semilunar valves. What comes first? Probably or argue any of them, but B is what we'll start with. It starts our systolic phase or systole phase. What comes next in of those four? We've got a signal from the SA node. What comes next? So next up would be A. You've got that signal bathing the top of the heart. That's going to hit the AV node. That's going to send a signal down. That's going to start what? Contractions. As soon as contraction starts, what's next? This one or this one? Contraction of the ventricles do what? Start to increase the pressure, which does what? Closes the AV valves first. Pressure continues to build. And then we get the opening of the semilunar valves, the emptying of the ventricles. So check valve closes first, can't go backwards, can't pump back into the veins. And then the opening of the semilunar valves empties the heart. So where are we going to find red blood cells carrying oxygen? Capillaries of the body, capillaries of the lungs, the right ventricle, or the left atrium? Where do we have oxygenated red blood cells? So what's probably the best answer up there? Is D. They're pure, definitely oxygenated. B, we're going from deoxygenated in the capillaries to oxygenated. So not every red blood cell in the capillary beds of the lung are oxygenated till they get to the end. Same thing with the capillaries of the body. They start out oxygenated, and then we drop all that oxygen out into the capillary beds. So by the end, they're deoxygenated. This is definitely, the right ventricle is definitely a wrong answer. So you could probably argue these two, but this is probably the best, purest answer there. So the sounds, heart sounds, lub-dub. Where do these sounds come from? A, the closing of the semilunar valves, then the AV valves. B, the opening of the AV valves, then the semilunar valves. C, the opening of the semilunar valves, then the AV valves. Or D, the closing of the AV valves, then the semilunar valves. D is our answer there. Which of the following sequence is correct? If I'm a red blood cell, which sequence is correct for the parts I'm going to flow through? Left ventricle, right atrium, lungs, pulmonary artery. Right ventricle, right atrium, body capillaries, vena cava. C, pulmonary artery, aorta, vena cava, left ventricle. Or D, body capillaries, lung capillaries, left atrium, aorta. Which is your best answer up there? Got 2 votes for B. Anybody else want to take a chance? or change your minds. On some level, they're all correct, right? I will eventually, with enough cycles, get to them in that order. The shortest cycle is number D. We go to the body capillaries, we come back to the right side of the heart. We go to lung capillaries, we go back to the heart, left atrium, left ventricle, and out the aorta. That's the shortest sequence. If I did this one, I got left ventricle, I have to go all the way through the body capillaries to get back to the right atrium. I go over to the lungs from there, and then I have to do a complete cycle again to do the pulmonary artery. So there's like 3 cycles in that answer. Left ventricle, I have to do a whole nother cycle to get back to the right atrium. They're not in order. So you have to do a whole cycle through the body to come back to the right atrium. We go through the lungs, back to the left side of the heart, body capillaries, and then come back to vena cava. So to throw them up there, I can make them all in order. It's just the question of how many cycles I go through to get them. But this is the shortest sequence to get all of them in order. So let's talk about sick cows. Edit this a little bit, so it might be different on your handout. We're going to talk about sick cows. Dealing with cows that go down and then possibly over. What are the problems there? What do we have to do? What do we have to do to take care of them? Acidosis and DAs. How do those things work? How do they occur? What happens when those conditions exist? We'll talk about a hypertonic saline IV and then diarrhea and dehydration in calves. So all of these are related to body water and rumen function. So how do we maintain the blood levels? How do we maintain the body water throughout the cow? So let's start with a question. Why can't we take ruminants to the International Space Station? I said they'll die of bloat. Does anybody know why? When we're thinking about our cows, in order for a cow's digestive tract, specifically the rumen and reticulum, to function, we need gravity as an element of that. The reason a cow only shortly lays over on its side and spends most of its time on its sternum, sternal recumbency, is we need that rumen to be oriented relative to gravity. We need liquid to fall down. We need gas to float to the top. And then in that last lecture, I talked about how that gas gets out. If that gas bubble is not there, it does not leave. So if you've got no gravity, the rumen is not going to separate like it should, and the cow's not going to be able to eructate to get that stuff out, and they're going to die of bloat. So they need gravity for that rumen to work. The liquid has to go down, the gas has to go up. And without that, they don't function properly. So we're thinking about that. A cow that goes down and over, we're now laying on her side. She's got the same problem she would have in the International Space Station. The gas bubble in the rumen is no longer in the right place. She can't belch out that. She can't eructate out all that gas anymore. When she tries to do it, what happens? Liquid comes out instead and ends up in her lungs. Normally what we're looking for, probably should go back to that diagram. So we have a sequence of mixing that needs to occur. And as things mix, move things around, we have this general, looking at from the left side of the rumen, a general flow toward the front. This is why all the heavy stuff ends up in the reticulum. And that's why we get hardware disease. Everything ends up here and then gets pushed through into her lungs. But we also have gas floating to the top, liquid floating to the bottom. So that diagram we gave you should be also be in your handout. You got that gas bubble up here at the top. We need to go through a series of contractions that are going to move that gas bubble from here down to the esophagus. And then they let that gas bubble out and they goes into their lungs, and then they breathe out that gas ahead of an exhale. So they kind of push it out of the system. If she's laying on her side, that gas bubble isn't where it is. You have liquid there, and she's going to bring that liquid into her lungs. So if we're looking at a down and over cow, they're laying on their side, we need to remedy that very quickly. So we need to get her upright. Job is to get her sitting against something. If she can't support herself, we need to bring in a big hay bale or put a hay bale in front of a skid loader and push up against her. We need to get that rumen upright. If she can't stay by herself, we have to assist her in some way. We're also talking about lifting her up on like every 6 hours, every 8 hours, so she doesn't get nerve damage in her legs. So getting the blood flow, getting the nerves back there. When our cows go down and over, they stop eating and drinking. Now they're going to be all right for a little while as the rumen's doing some things, but eventually it's going to run out of energy, run out of protein. So we need to provide that animal with something to eat, something to drink. So certainly eating, we can lay stuff in front there, usually with the 5-gallon buckets that we're providing. She's going to knock that over more often than not. So it's more of, we got to bring it in. She drinks, take it back out, and do that on a regular basis. If we're going to get that animal back up, she needs energy. She needs liquid to stay hydrated. So we got to get her propped up. We got to get her eating and drinking, and we got to do what we can to eliminate that dehydration that's happening while she's laying there. Your average cow is going to go through about 30 gallons of water a day. So that's 6 5-gallon pails. Making sure she has access to that water while she's stuck there on the ground is very, very important. So rumen orientation and function. We're going to need feed and water to make sure that rumen functions the way it's supposed to. And then it's going to have to be upright, vertical, next to gravity. So the air pocket, the gas pocket, the carbon dioxide, methane, and sulfur dioxide that float up to the top are where they're supposed to be when that reflex comes through to get rid of that air out of the rumen. We also, if we're going to mix right and keep things flowing the way they're supposed to, we need that vertical orientation. So down cows, either getting them standing up or propping them up with a hay bale, something soft that they're pushed against, and then something solid that they're not going to get, not push away. A little hay bale might work for 20 minutes or so, but then she's going to knock that over. So having something behind that bale that's heavy can help as well. So moving over to acidosis and DAs. So acidosis, everybody here know what acidosis is? Okay, we got one no. Anybody else have any ideas? So we have bacteria in the rumen. They act independently of the cow and not always in the cow's best interest. If we put enough fermentable carbohydrate in the rumen too fast, those microbes are going to ferment that and overdo it. They're going to create more volatile fatty acids, taking the carbohydrates, breaking them down into acids than the cow can handle. So acidosis is an overload of acid to our cows. So if we're looking at a typical cow, typical fermentation, this was part of your lecture last week, blood we'll say is around 290 milliosmoles. Usually the rumen is down around 260. So what do we have up here? In the blood, in the body, that 290 is what? What kind of tonic? It's hyper relative to the rumen, but that is in the body, it's always isotonic. So you have isotonic out here in the tissues, and normally you have hypotonic in the rumen. The rumen actively takes the VFAs, the electrolytes, and moves them over to the blood. This is one of the cases where water does not follow solute. The rumen lining actively moves those electrolytes, actively moves those VFAs, but leaves the water behind. There's that wall in there, a lining in there that leaves the water behind. We need water in there. So this is normally hypotonic. The water wants to push out, but it can't. It's not allowed to. So the rumen is designed to keep water in and not let it out. I think this is the redneck hot tub lining a pickup truck, right? So if we line the pickup truck with plastic or tarp or something, we put the water in, the wall of the pickup is going to hold that water back. The water wants to go out, but the pickup's going to hold that back. So that's the way things are normally. What happens after a very fermentable meal? A cow gets loose, gets in a grain bin, eats a whole bunch of high moisture corn, a whole bunch of grain. On an empty rumen, you get tremendous fermentation, which generates lots of VFAs, which are all solutes in solution. They're osmotically active. We get an overload of solute and we get hypertonic relative to that isotonic blood. So that's going to make the water want to go this way. And that's not normal. The body is not designed to stop that flow of water. Just like that redneck hot tub, if I can get behind that plastic, I can put my finger right through it. Whereas going the other way, I can't get through. But if I can get behind that plastic, I can put my finger right through or a pin or anything there. So water is going to start to flow in the wrong direction. And this has a tremendous set of consequences. So we have acute acidosis, which is a cow gets loose, eats a whole lot of grain, a whole lot of wheat, something, and fermentation goes nuts. We generate a lot of VFAs. Those all add solute to the water. They make the rumen hypertonic. So you're looking at something at 350 milliosmoles or greater in the rumen. And that's going to lead to all kinds of problems. So if water were allowed to move freely in this scenario, which it starts to do, we run into problems. So remember this diagram here? We've got the 65% of the total body water is inside the cells, 20% the GI. We split that out to 13 and 7. We've got plasma and then other extracellular fluid like the pancreas, like saliva, all those things that in the cow. You've got 8% in the blood, 13% in the rumen. If we want to try to equal that out on a 1,300-pound cow, the body is going to, or water is going to try to reach equilibrium between the blood and now this hypertonic rumen. So in a 1,300-pound cow, you're looking at 106 pounds of water in the rumen, the reticulum. You got 68 pounds of water in the plasma. If I want to take that ruminal solution from that hypertonic back to equilibrium with the blood, 300, 290, I have to move 24 pounds of water to do that. 3 gallons. So I'm taking 24 pounds out of here. That leaves me with 44. She's going to be in trouble from a blood volume standpoint. And this is what a cow with chronic acute acidosis getting loose in the grain bin dies of: dehydration. All the water flows to the rumen to try to equal things out. And she doesn't have enough blood volume to get oxygen to her body. Now, this is rare. Not every cow gets loose in the grain bin, but we have little times throughout the day where we've got subclinical acidosis, where we do little insults every day. So if this were allowed to happen, and it does, because we can't stop that flow, we get large swings in the water pools of the body. You've just expanded the rumen to 140, and the blood's down to 44. So that's not a good thing. So we're looking at acidosis. We get that rapid fermentation. I forgot to add into your things there. With that increase in VFA, which are acids, you're going to get a tremendous drop in ruminal pH. So you can add that over on the side of your handout there. But what we're concerned about is the solutes in solution. So increased osmolality, increased solute, those VFAs are ruminally active, osmotically active. Water starts pushing into the rumen to help equalize things out. You're going to get damage to the lining as it tears through. Water's not supposed to flow through. So busting through and getting into the rumen is going to rip holes. It'll start out as blisters that form on the side of the rumen. And then those blisters will start to tear. Now we have openings in the rumen. Holes in the rumen. Does that sound like a good thing? Peritonitis is coming our way. So it's going to dehydrate that animal. You're going to get damaged lining. You now have open holes to the inside of the cow. Anybody here smelled, handled rumen contents? Nasty, nasty stuff. You don't want that in your bloodstream. Full of feed particles and bacteria. So we have holes that are going to leak water and room. So water is going to be coming in, contents are going to be flowing out. All of that leakage is going to go to the liver first. Does anybody know how it gets there? Goes into the blood, the capillary bed that surrounds the rumen. Then where does it go? All that blood is collected into the portal vein. And then we open that portal vein, opens up into a capillary bed that feeds the liver. So all of those leaking contents are going to end up in the liver. So if you've got a cow with subclinical acidosis, little leaks over time, you're going to start to get liver abscesses. As contents, little bits of contents end up in the liver, they're going to become infected. That damaged lining becomes scars. And we're going to say never absorbs again. The studies that have been done on this know those scars persist until the next lactation. Do they last forever? Don't know. Nobody's measured that, but we're going to assume they do. At least I got scars on my hands that have been there a long time. So once you've damaged that lining, it can't absorb VFAs anymore. What does that predisposed the animal to now. We've reduced the absorptive surface because we've got scar tissue there. VFA doesn't get absorbed as fast, and you get backups faster. You get hypertonic faster than before you had a healthy rumen. So those scars never absorb again. And what we're talking about usually with our dairy cows is subclinical. They got a couple hours every day because we're not adjusting our dry matters the way we should. We got too much grain in the diet. Our balance is not where it should be. We got periods of 3, 4, 5 hours where they're fermenting too much. And the VFA spike, the pH drops, and that happens for a few hours. They don't feel too good. They back off on intake, and then they'll come around and continue to eat. They look all right. They're not going down. They're not getting there. So acute or chronic, those are clinical signs. Acute is when an animal gets loose in the grain bin. It has 20 or 30 pounds of grain that they're not supposed to have at one time. So growing up in a barn, we'd always put the chains up across the cross alleys at night so the animals could not get into the grain room. Chronic is what you're going to find in a feedlot. They're keeping those animals on a high carbohydrate diet. They're right on the edge. They're trying to get as high gains as they can. So they're right on the edge. And if they don't manage their feedlot really well, they're going to fall over into acute acidosis. So it's about keeping those rumens full, making sure that whenever they're hungry, there's food available. So they do lots of little meals throughout the day. And for whatever reason, a feedlot bunk gets empty and those rumens empty out. If they do a feedlot diet and eat that back to full, their full gut, they're going to jump into acute, and then they're going to have all the problems associated with that. So acidosis effects on the cow. So if we're looking at an acute situation, cow gets loose in the grain bin, you're going to have high VFA, which is going to lead to high osmolality. The rumen becomes hypertonic and wants to go the wrong way. It's really strong about keeping stuff in. It tries to push out. It doesn't let it, but it's not strong the other way. So if water wants to come into the rumen, it starts to bubble up the tissue as it fills up, forms blisters. Like any blisters, eventually they're going to pop. And now you have a hole in the roof side of the lining. Now water is moving freely both ways and contents leave. You've got feed leaving, you've got bacteria leaving, you got protozoa, fungi leaving. They're all going to be collected in the capillary bed of the GI. They're going to go through the portal vein, then to the liver. Liver's going, well, I don't want this stuff here. So we start to try to filter, we start to try to detoxify. So the liver is trying to do what it can to destroy that bacteria, to destroy that feed. So we get an inflammatory response by the liver, and that's going to spread to the entire body. So there's a massive release of histamines. Anybody here take antihistamines or allergies or things like that? You're trying to keep the swelling down in your nose so you don't sneeze. So we're trying to counteract the inflammatory response. The inflammatory response is meant to draw white blood cells to a particular area to help fight off an infection. So you got the liver responding with an inflammatory response. There's a lot of nice work about that done out of Iowa and Michigan State. Then abscesses from the bacteria and feed particles start to form in that liver. So the liver is no longer going to function the way it's supposed to. And our dairy cows need their liver functioning at a high level to make sure that they can produce the milk that we need them to. Those histamines are going to leave the liver. They're going to go to the foot. They're going to go to that corium area. What are they going to tell the corium to do? Along with every other tissue in the body, what are they going to tell the corium to do? Inflame. What happens when the corium inflames? Starts dying off because it's between two hard things, the bone and the hoof. Can't inflame anymore. So you're going to get laminitis, you're going to get clots from dead tissue. That's going to look like this. So the rumen is going to start leaking. So you've got bacteria, fungi are collected in the capillaries. They're going to go in the portal vein. They end up clogging the capillaries of the liver, the bacteria, the feed particles. They're going to send out that inflammatory response. Got the blood from the liver going back to the heart, to the lungs, back to the heart, out to the capillary bodies. We start to get inflammation of that corium that we call laminitis. So with that laminitis, what's not going to work anymore? Probably the formation of hoof. You're going to lose lower quality hoof formation. So cows that are chronically overdoing it or have little bits of subclinical acidosis every day start to get curly toes because this wall weakens and it starts to collapse. You're not going to get good growth underneath anymore, so you're not going to have a strong protection from the surfaces on the bottom. You're going to have a bunch of animals that look like they're walking on broken glass. They're all going. I've seen entire herds that way. So everybody following acidosis. So that's why something that happens in the rumen affects what the feet look like. You're also up on that coronary band. You're going to see all your cows. They're going to be bright red and swollen. It's also going to impede blood flow as well. So if they got laminitis, you get kind of a backlog of blood around that top of the hoof. So continuing on with DAs. DAs are, in my mind, related to acidosis. Displaced abomasum, twisted stomachs. This is a theory of mine. I don't know if everybody agrees, but when I presented this to my PhD committee, they're like, yeah, that makes sense. So we'll see if you agree with me or not. So displaced abomasums. We have a abomasum, that fourth compartment of the stomach, fills up with gas. And what do things that are full of gas want to do, especially in a liquid environment? They want to rise, float up. And what usually happens is it twists one way, right, or it twists left. So if it twists, we're not going to be able to get feed into it. We're not going to be able to get feed out of it. So the problem with the displaced abomasum is the gas is coming from microbes that are still alive in the true stomach, and they should be dead. Does anybody want to guess why? The acid from the stomach should drop the pH to 2, and they all should die. But if they're producing gas, that means they're still alive. So question is, why are they still alive? Causing problems that we don't want to. So there's a certain sequence of around calving that, to me, promotes these displaced abomasums. Now, anybody who's tried to do any research with DAs, well, we're going to do the perfect diet, small particle size, lots of starch, and all these cows are going to get DAs. It never happens. They try to make it happen, they can't. So we don't fully understand exactly what's going on. But when we're calving, we've talked about hypocalcemia. Why does that occur? We're not pulling that calcium out of the bones fast enough, right? So we've got hypocalcemia. That's going to lead to what in the muscles? Muscles going to work? No. So certainly the GI tract is full of muscles. Those smooth muscles are the first ones to be compromised. So if you have hypocalcemia in your abomasal compartment of the stomach, what is that going to be like? Is it going to be toned anymore? No, it's going to be sloppy and loose. When we switch our cows over from a dry cow diet to a lactating diet, we're increasing the nutrient density. We've got more carbohydrates. We got more protein. We're generating a more fermentable diet. If that animal is not ready for that, she's going to get a short period of acidosis. Acidosis is going to overwhelm the rumen, and you're not going to be able to absorb those VFAs as quickly as you might. So normally we want from the rumen, the reticulum, and the omasum, we want all the VFAs absorbed before they get to the abomasum or true stomach. VFAs, if you get too many in the abomasum, they're going to act as a buffer. They're not going to allow the pH to drop. So if your close-up diet's not prepping her the way it should, she's overdoing it when she first gets on a lactating diet, she's going to get all kinds of VFAs to the true stomach. When the acid is secreted by that true stomach, those VFAs that have overloaded the system are going to prevent a pH drop. That prevention of pH drop allows those microbes to continue to live. They continue to ferment. So they're producing gas in a loose, floppy stomach that's kind of twisted off. So there's nowhere for the gas to go. So we're producing gas where we shouldn't. We have a digestive tract that's not contracting like it should. That might push that gas back up the tract, and it goes out in normal burping. But we don't have any tone anymore. So those two things together, the little bit of acidosis, a little bit of hypocalcemia, seem to me are going to predispose that animal to a displaced abomasum. Don't have tone. We got gas production because we're not killing those microbes. So a lack of tone, too much VFA. Those two circumstances in the abomasum, you got loose and sloppy. Bacteria are not killed. You got the production of gas by the bacteria. That abomasum fills up with gas and twists one way or the other. Like I said, that's my theory. Does that make sense from a stepwise step fashion? So we have circumstances here, but they certainly follow a logical sequence to lead to that displaced abomasum, that twisted stomach. But as we all know, logic is the art of going wrong with confidence, right? So it makes sense. It's a theory. And that to me is why we got to make sure we've got no hypocalcemia in our fresh cows, no acidosis in our fresh cows. Hypertonic saline. We're putting hypertonic saline into our cows. Does anybody know what vein we usually put it in? Which one do I want you to remember? External jugular. So that's where we put most of our IVs. If worse comes to worse, find a milk vein. There are some people that are like, you really don't want to do that. And I'm like, well, it's between her and dying. I'm going to go for it. I think the reasoning behind that is we don't want to scar up the milk veins. But to me, they're a vein's a vein. Let's get it in there. So we're putting that hypertonic saline in. So what do we know about that solution? Makes them want to drink, but how we describe that solution? Got an osmolality, I think, of 1500. So it's way more than isotonic, like 5 times more than isotonic. So it's got a lot of extra salt and chloride in there. And we're going to throw that into the vein going back to the heart. That's going to go through the heart to the lungs and then out to the body. You're going to have an increase with that hypotonic saline in your system. You're going to have an increase in osmolality and an increase in sodium, both of which are sensed by the brain. Has anybody done this, given a hypertonic saline to an animal? You give that animal, you usually want to have 2 5-gallon buckets of water right there. You give that IV, she starts drinking within 2 minutes. And I mean, aggressively drinking. So why are we doing something like this? We're trying to get as much water into her as we can to help hopefully flush the body of toxins. How long would it take us to get 10 gallons in there through an IV? Probably days, right? This is a way to get a whole lot of water in as opposed to just doing isotonic saline solution. We get a whole lot of water in there. So the osmolality goes up with the blood. That's wrong. The sodium goes, that's wrong. The brain says, I need water, I need water, I need water. This is why they always have salty foods in bars, because they want you to drink more. Have a very strong desire. We're talking about consuming at least 5 gallons, maybe 10 immediately. Those 2 5-gallon pails are usually gone. So the brain says, I need to drink and I need to drink fast. That water is going to flow through the gut first. That's going to flow out into the blood and eventually get to the kidneys, where you're going to have a lot of water coming out the other end for that animal. 10 gallons is a lot to consume in 5 minutes. That's 1/3 of what they usually drink in a day in 5 minutes. Instead of 8 hours, 5 minutes. So the idea is we got the water going in. It's going to go GI to the blood, to the kidney, to the bladder, and out. We're trying to get a wave of water through that cow. I've got all that water in there. That's going to help me get that sodium and chloride back to where it's supposed to be, dilute things out. Hopefully with that, it will flush the toxins that are not recognized by the kidney out faster. If we were counting on her, if we've got toxic mastitis, for her to drink, she's not going to do it. She doesn't want to. I don't feel good. I don't feel like drinking. But the sooner we can get those toxic compounds out, the better she's going to feel. This is one of those cases with toxic mastitis that actually the white blood cells doing what they do, the phagocytosis, the killing of the invader, actually makes things worse because the contents of the bacteria are now spread. You've blown that bacteria up. you've spread all those toxins throughout the body. And the first thing that does is, I don't feel good. I don't want to eat. I don't want to drink, when that's exactly what she needs to do. So we're forcing her with this hypertonic saline through that portal vein to flush the stuff out of her body. And we'll talk more about how that works when we get to the kidney lectures in a couple weeks. But that's what we're trying to do. Justin up in the dairy is like, is there any harm in giving this to a cow when you're really not sure what she's going through? I told him, and I'm not a vet, so I probably get in trouble by some vets, that if you've got enough water, there should be no harm with this. You got to make sure that she's got enough water to drink for the next few hours. So he goes, well, what if I'm not sure if it's, she's toxic or not? Can I just give her an IV? I said, as long as you got water, you should be fine. So hypertonic saline, we're trying to get her to drink water because it would take forever through an 18 gauge needle or 20 gauge needle for an IV. And you don't want to sit there with a cow with an IV for 10 gallons. You can do it, but it's not. Cows get tangled up. They start ripping their IVs out. This is a faster way to do it. Diarrhea and dehydration go hand in hand with our calves. So 2 major causes of diarrhea in our neonate calves. We can have damage to the lining of the intestine. We lose the surface area and function. You can't absorb nutrients anymore. And as long as those nutrients stay in the lumen, the body's going to try to equal things out. So if stuff does not get digested and absorbed, it stays in the gut. Water gets drawn to it, and we get diarrhea. We're not getting the absorption that we should. So transport the water through the intestinal cells into the lumen of the gut. We sometimes have situations where we've got toxins or poor energy where things are being pulled into the gut. So you've got either a destruction of the absorptive surface area, so we're not absorbing, things are moving through the gut, or something in the lumen of the gut, and the inside of the gut is drawing water out of the body into the gut, and that's why we get diarrhea. So diarrhea is going to lead to dehydration. So dehydration in calves, we've got water in, water out. The water going out of the body exceeds the water coming in. We start to lose those water pools of the body. So blood is going to be defended. The volume of blood is going to be defended at the expense of all others. What might be an advantage for that? What happens if I don't defend the blood volume? I steal blood, liquid, water from the blood and maintain my other ones. Can't get oxygen to the tissues anymore. The highways of the body shut down. So interstitial, intracellular, extracellular are all going to be used up at the expense of the blood pools. So we're losing or becoming dehydration. What are we losing? Going to lose water. What else? Electrolytes. Water and electrolytes. So if we're going to rehydrate that animal, we have to do water and electrolytes into their body. Bringing in water is going to mess, just water is going to mess them up. So we need to get electrolytes as well. So how do we feed a calf to replace water and electrolytes? So usually we have some sort of product that's going to provide the electrolytes. We dilute it with the water we have at our barn, and we feed it to that animal. We want to feed it separate from milk if we can, because milk, there's some places, well, you just mix it in the milk, everything will be fine. But if I put the water and electrolytes into the milk doesn't clot the way it's supposed to in the gut and doesn't stick around and just flows right through the calf. Anybody ever seen that? Goes in, you feed the cow, and 2 minutes later it's coming out the other end. Things are that damaged in that calf. So if you're looking at the ingredients, you're going to have sodium and chloride, electrolyte, potassium phosphate, potassium's electrolyte, citric acid, citrate. They're going to act as energy sources. So you're trying to get your electrolytes in there, sodium, chloride, potassium, and hopefully a little bit of energy. You want to feed it separate from the milk, usually a couple hours difference if you can. I think it's probably better if you do the electrolyte solution first and then come in with the milk afterwards, allowing that milk to do all the processes it needs to, whereas the electrolyte solution can go through and get it done when it needs to get done fairly quickly. I think if you do it the other way, sometimes that electrolyte solution will come in and interfere with that milk digestion. So a little closer view there. Same sort of thing here. You've got sodium chloride, potassium, your electrolytes there. Lactose is going to be your energy source. So contains lactose, electrolytes. Citric acid can be an energy source, but it's also probably acting as a preservative in that case. So what happens if you don't have a commercial product on hand? What do you do? You can think about any of the number of things that are out in the real world for us. Gatorade, Powerade, it's better than nothing. Probably too much energy for these animals. If you go with the G2, that's low in sugar, probably better. There's a lot of homemade recipes out there. So we've got low sodium salt, baking soda, that's going to get bicarb and sodium in there. Some pectin to slow things down, a little beef broth in there. So same sorts of ingredients moving through. This is just pure electrolytes in there. So you got table salt, sodium bicarb, and then salt substitute, sodium chloride. So lots of things out there. Here's how to make your own Gatorade if you're in the mood for it. But get some water in there, get some electrolytes in there. She's down. That means you got to feed her more water than you normally would to get her back to where she needs to be. So electrolyte solutions. It also can be helpful to throw some B vitamins in there to help process, to help get her back to where she needs to be. B vitamins do not stick around in the body too long. So if an animal gets sick, adding some B vitamins in there can be helpful. So why don't we stop there, take your break. We'll start again at 2 after on that clock. Need another sheet. Yes, please. It's right in there. Okay, coming back. So your letters. Anybody had a chance to study their arteries and veins yet? Let's try a couple of them here. So pulmonary artery, what does it do? So it's an artery, what's it going to do? It's going to drain or deliver? Deliver. So it's delivering to something, going away from the heart. So your choices are delivers to the blood to the lungs, delivers blood to the rear or back leg. Pulmonary is what? Lungs. Pulmonary artery feeds the lungs, delivers blood to the lungs. Abdominal aorta. What is that going to do? Aorta is what? It's an artery, right? So it's going to deliver blood somewhere. Where do you think it's going to deliver blood to? The abdomen or abdominal area. So gastrointestinal tract is certainly part of that. A. It also probably delivers blood to the kidneys and liver and all that stuff as well. Common carotid artery. Everybody know where that one is? As you get older and we have a lot of people dealing with clogged carotid arteries, they have to be rooted out. That's gonna, this in the neck here, carotid. So it's gonna feed the head. So artery is gonna feed the head, deliver blood to the head. That's C. Brachiocephalic trunk. What does that do? Delivers blood. So you can guess from there, what is the brachiocephalic trunk? Is it an artery or a vein? Based on the answers. Artery. So the question is, where does it deliver blood to? Brachial means what? Wrist? Forearm? Cephalic means what? Head. So C and D would be the proper answers. Either one of those. External pudendal vein. What does that do? It's going to drain. What do you think that does? Nothing really there to help you, right? It is B. It's going to drain the mammary gland. You have a right and a left one. We're just going to worry about the generic one from either side. Portal vein. What does that do? Portal vein. Where did we say that was today? Talked about it in acidosis. It's going to drain the GI tract. So that'd be A. Cranial superficial epigastric vein. What's the common name for that? Yep, the milk vein. It's the one on the gut. So cranial toward the head, superficial on the gut, epigastric out on that belly. So what's that going to drain? B, your mammary gland. So we have two different veins that drain the mammary gland. Cranial vena cava. That's the last vein before we get to the heart. So what is that going to drain? So any of the answers B, C, or D are correct. So you have the cranial the milk vein. It's going to drain into the cranial vena cava. The head's going to drain into the cranial vena cava and the front leg are all going to drain into that vena cava. So you've got cranial, caudal, superior, inferior vena cava moving through. Any questions on those? Got a little work to do, right? We got you started here today. So let's talk about the lungs for the 15 or so minutes we have left. So switching gears, we're going to talk about the lungs. So this here is Taffy. It's probably before your time, but she was the fastest cow in the world in 2001. I was there at Michigan State when she got that record. Taffy was part of a research trial where we were trying to exercise dry cows to see if exercise made a difference in how they transitioned into milking. Taffy was one of the initial goers. They took her ovaries for another experiment, and they said, well, we got this cow here. We can either beef her or put her on this. Let's see if we can train a cow to work on a hot walker. So anybody here equine know how hot walkers work? We hook the horse onto it. It moves at a steady pace and pulls that horse around. You think that's going to work with cows? No. So they're just going to plant their feet and do that. So the hot walkers tend to be open. It's a track on the ground. What was created was a series of gates that made a circle and an alleyway in where the path should be going around. And then we dropped down electric fence coming up behind her to keep her moving forward. So we exercise these cows every other day. It was Jill Davidson. I think she works for Purina now. That was her PhD. We train those animals and then we put them through a stress test. Anybody here have anybody in their family have a stress test or gone through a stress test? What is that? Put you on a treadmill till you can't go no more, right? So we put these cows on a horse treadmill and we took blood more or less continuously, measuring lactic acid levels. We're measuring respirations, and we just kept that thing going faster and faster, tilting it up until they couldn't do it anymore. Taffy was an outlier. She was a machine. She just kept going and going and going and going. So when we're training cows out at the dairy, so word gets around the department. This is Brian Nielsen. He said, well, I bet I could train her. He's the equine guy at Michigan State. So when Taffy was sold at auction, he went there, bought her. She had to be sold at auction because everybody in the state of Michigan has to have access to it. So he bought her, took her back to, he's an equine guy. He's got his home track laying out there, and he trained her with a saddle and everything to run. And the inaugural Mid-Michigan Cow Racing Association, she was one of the few cows that finished the race, and she went about 6 miles an hour. So that's a pretty slow trot, but just because she was under control and knew what she was, she became the world record, the fastest cow. So part of that is she had an incredible set of lungs. So she could bring that oxygen in and just keep going and going and going. So we know her there. When I post these are the articles related to that, the YouTube video showing how that happens, that first inaugural race. Since then, her record's been broken a couple times, but she was the first one to get on there. And Brian Nielsen got on all kinds of talk shows. I think he should have brought Taffy with him, but he was the world record cow racer for a while, and he got on to Tell the Truth and a couple of late night shows and things like that. So thinking about Taffy, she had a tremendous set of lungs. So let's think about how cows breathe. When we're going through this, we're going to answer this question: how do cows breathe 4 different ways? One is we're going to talk about the parts of the lung, how everything works together, how things go in order. We're going to talk about how breathing works. How do we inhale? What muscles are involved? How do we exhale? What muscles are involved? We'll talk about the lung volumes and capacities. We assume that Taffy had a tremendous lung volume and could expand her tidal volume way faster than most other cows. And then we'll talk about the carbon dioxide and oxygen exchanges that occur in the lungs and occur in the tissues. So we're going to answer the how do cows breathe 4 different ways. So let's start with the parts. How do cows breathe? And we'll get you the handouts. They're not back from the printer yet, but you should be able to keep track of this pretty straightforward. So lungs are in the chest area between the shoulder blades. We have the entrance to those lungs up here. We have the delivery system, the trachea, the larynx, gets that air down inside the lungs. And then we kind of have that balloon that is the lungs that moves in and out, breathing through those passageways. So using our atlas of bovine anatomy, we're going to have the entrance is either in the nose or the mouth. Air comes in. We go back through the soft palate. We go to the larynx, the pharynx, and then we get down into the parts of the lungs. So the first part of the entrance into the lungs is shared for a number of different purposes: vocalization, eating, drinking, and also air exchange. So we have that common beginning, and then we move over to our lungs. So if we look at our lungs, we have a picture from our 8th edition of the Atlas or Anatomy and Physiology of Farm Animals. We got the horse, a little bit bigger than the cows. If we zoom in on that ox or that cow, the lungs are going to go from the first rib to about the 13th rib in our cows. You've got the heart sitting here, and that's going to displace some lung volume. You're going to have 5 different lobes on this lung. And that's going to be where we get oxygen in and carbon dioxide out. So we're looking at, again, the Atlas of Bovine Anatomy, dorsal aspect of the bronchial tree. So what's happening from a looking perspective? What are we doing? Dorsal aspect. Nope, we're looking down, standing on top of her, looking down on that. So this is the left side. This is the right side. The heart sits up in here, takes that. So we got 5 different lobes, 1, 2, 3, 4, 5. There, each fed by a series of bronchi, bronchuses, and bronchioles. So we start out with the trachea, the common singular there, something that's rigid, something that does not collapse as the lung and the animal breathes in and out. And that starts to branch off into still rigid kind of containers. They have a little bit of give. That's why we get problems with asthma and things. They can open up and close down just a little bit, but they're still fairly rigid. So we go from the trachea to the bronchuses, singular, bronchi. So 1, 2, 3 branches that we start with, and we proceed down to smaller and smaller bronchioles. So getting down in there, mostly rigid pathways that are always open, no matter whether we're inhaling or exhaling. That rigidity makes sure the air can move in and out. If they opened and closed, that would cause some real problems. So this, what we're looking at here, these 3 spaces, trachea, bronchi, and bronchioles, these are the transport areas of the lungs. They're meant to move air in and out. So if we look at a human here, we're looking at the human chest. This is the left side, the right side. The heart sits right in there. We have the same thing, the trachea, the bronchi, and the bronchioles working down through. Eventually, we get down to the alveoli at the end. This is the terminus of where the air ends up. The trachea, bronchi, and bronchioles are mostly rigid. These alveoli can move in and out. They expand and contract. When I inhale, these get larger. When I exhale, they get contracted. Taking a look at, again, the human, we zoom in. You got these alveoli down at the end of the bronchioles, all these little air sacs. They're going to be covered with that net of capillaries we talked about during the pulmonary loop. So coming in, got these alveoli having a net of capillaries around them. I think the next picture is a little bit, nope, it isn't. So you've got, this kind of shows you're going to have a artery coming in, goes into that net of capillaries. Those capillaries are going to end up in the, nope, got them backwards. Blue comes in, artery, the red going out is the vein. At least it should be from a color perspective. Because oxygenated, non-oxygenated is coming in, oxygenated is going out. So I think they got that backwards on that diagram. This is a better kind of look at it. So when this one's labeled correctly, you've got the blood coming in, you've got that net of capillaries, one on each of the alveolar sacs, and then we have the drainage going out, oxygenated blood going back in the veins, back to the heart. So we're talking about parts and breathing. We've got the nose, mouth, the pharynx, larynx, trachea. You've got the bronchi. They have different parts. We're not going to worry too much about those. And then we get down to the bronchioles at the end. At the end, we have the alveolar ducts and the alveolar sacs. So working our way through, we come in the mouth, go to the larynx, trachea, bronchi, bronchioles, through the ducts, and into the sacs. The top here is involved with transfer of air. There's no exchange of gases between what's inside and what's in the tissues. The ducts and the sacs are where the air is exchanged. Carbon dioxide removed from the blood, oxygen put back in. So these are very thin. They're very elastic. They get bigger and smaller. This is where the gas exchange occurs. These tend to be rigid, and we just allow for the transfer of air through them. So that's probably a good place to stop. So those are the parts associated with breathing.